This is a video about cell division and special kind of cell division, which is reproductive cell division, or other name meiosis. I highly recommend to download the text file which with the link in the description, and probably presentation. It will save time, and also you will remember the names of these parts are more clear. Okay, first we will pay attention to organelles which participate in the cell division, and this is the nucleus, of course, with nuclear membrane, and centrosomes here. So, meiosis takes place in an organism reproductive structures called gonads for the sole purpose of producing haploid gametes that are genetically different. These haploid gametes are sperm cells in the male and eggs in the female. In plants, pollen grains contain the male gamete, while structures called ovules contain the female gamete. So we have male testes, male gonads, and this is testis, and ovary, it's female gonads. Here is more with more details. We see that testis and how this spermatids are produced, or male gametes. In female, it is different, it's ovary, and we see here with some details how the ovum, the egg, is produced. So, an original cell that undergoes meiosis will have replicated its chromosomes during interphase of the cell cycle, the same like in mitosis. This original cell is diploid, which means it has two sets of chromosomes, one from each parent, sometimes written as 2N. Through two stages of cell division, meiosis produces four genetically different haploid gametes, sometimes written as N. For this reason, meiosis is actually called reduction division. So not only reproduction, but reduction division. It reduces the total chromosome number in half. So when haploid sperm cell and haploid egg cell unite to form a zygote during fertilization, the diploid number of chromosomes is restored in the resultant zygote. Okay, we have 46 chromosomes, and they are paired. So we have in total 22 autosomes and 2 sex chromosomes. In male, it is X and Y chromosomes. Here, that's a short presentation of the stages of the meiosis. We'll go into details. And the first stage is interphase stage, when the chromatin fibers in the nucleus are replicated. So the number of these chromatin fibers duplicate, but also what duplicates? Centrosomes. Okay, let's go. Meiosis is broken down into two stages of cell division called meiosis 1 and meiosis 2. Meiosis 1 has four phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2 also has four phases: prophase, metaphase 2, anaphase 2, and telophase 2. Prophase 1 starts with the diploid cells. Okay? Its DNA in the form of chromatin fibers contain two sets of chromatin fibers, one from each parent. Altogether, it is 46 sets for humans. After the DNA in the form of chromatin fibers replicates, it condenses into the X-shaped chromosomes that are made from two identical sister chromatids of V-shape. So every chromosome now is replicated, and we change name to chromatid. Two identical sister chromatids, and they produce X-shaped chromosome. Okay, we see here interface. During the interface, centrosomes duplicate, and chromatin fibers duplicate, and what we get in difference from mitosis, that the homologous chromosomes pair and produce tetrad. Tetrad, it is a couple of two homologous chromosomes, and also every this X-shaped chromosomes consist of two sister chromatids. We say chromatin is fiber, chromatid is V-shaped chromosome, and tetrad, and then tetrad is combination of two X-shaped X-shaped chromosomes, or its other name, tetrad. Okay, we will go further, and as I said, we have chromatin fibers, and they condense, they replicate and condense into X-shaped chromosome, which further will be combined in homologous chromosomes. They group together, pair together. So, and centrosomes with centrioles inside, they also duplicate. Here we see during the prophase 1, homologous chromosomes pair up and exchange fragments. So what is again very much different from mitosis, that we have a special process which is crossing over, and this is exchange of genetic information between homologous chromosomes. So let's jump a little bit to this picture. So we have X-shaped chromosomes from two sister chromatids. Every chromatid is actually chromosome, but when they're paired, they change name to chromatid. So then we have paternal X-shaped chromosome, and maternal chromosome, X-shaped chromosome, and they produce together tetrad. Pardon. Okay, they produce tetrad. Here we see it. Okay, next, what is, as we know, this is a homologous chromosomes. They combine in pairs. In difference to mitosis, sister chromatids in these X-shaped chromosomes, they are in mitosis. From centrosomes, such fibers, microtubules, they are connected to centromere. Centromere, this area which two V-shaped chromatids are glued to each other. But this is in mitosis. In meiosis, it is different. Meiosis is different. Let's go. So chromatid pairs in the dyad are normally genetically identical and said to be homozygous. However, if mutations occur, they will present slight differences, in which case they are heterozygous, different from genetic code of parents. Paternal and maternal dyads, that are pairs of chromatids, group together into tetrad, which consists of two dyads. Then genetic material from the paternal and maternal copies of each chromosome is crossed over, creating new combination of genetic code on each chromosome. This stage is called synapsis. So we have two sets of X-shaped chromosomes, homological, they are coming together. What is happening next in synapsis? There is exchange of genetic information from one homologous chromosome to another. So it's schematically it is presented like here. And it can be single crossover or even double crossover. So you see these chromosomes exchange the swapping of genetic information, and they produce different genetic code. Okay, so let's read a little bit. So in a process called crossing over, chromatids from each homologous chromosome exchange genes that are segments of alleles. So it is some genes which are responsible for alleles. Crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. The crossing over results in genetic variety or in offspring. This is why children are different from their biological parents as well as from their biological siblings. The number of combinations after this crossing over is millions and millions, so siblings always different. Here again, schematically presented that two dyads combined in tetrade, and then during crossing over process, they exchange some parts of genetic code, this swapping, and then we have in total all these V-shaped chromatids. They have different genetic code. Number of combinations is at millions. And this is why siblings look so different from each other and from parents. Little bit resembles, but mostly different. Chromosomal crossover or crossing over is an exchange of genetic material during sexual reproduction between two dyads. Crossing over of the chromatids results in recombinant chromosomes that are different from parents' chromosomes. Due to the swapping of genetic material during crossing over, the chromatids that are held together by the centromeres are no longer identical. A part of the chromosomal crossover, there is a sister chromatid exchange, that is exchange of genetic information between two sister chromatids. It can occur, sometimes it occurs during mitosis or meiosis. Why? And it can be a part of the process to repair the damaged mutated DNA. Okay, then continuing on prophase 1, the nuclear membrane disappears. The centrioles, now we have two centrioles, they move to opposite ends of the cell and spindle fibers grow out of them. Next, in metaphase 1, the tetrads line up at the equator and attach to spindle fibers from opposite poles. Okay, so let's look. Here we have metaphase. Metaphase, we have these spindle fibers or microtubules grow from centrioles, and every fiber connects to this dyad, cis dyad or X-shaped chromosome. They can be paternal, maternal can be swapped, so it is up, down, it can be different. And as I said, arrangement of the spindle is random. And during this stage, they move, these tetrads, they move to the center of the cell between the two centrioles. Okay, next stage, anaphase, when these spindle fibers or microtubules, they pull every X-shaped chromosome to each other and separate them to one half and another half of the cell. Okay, so let's look again. And so we have interphase. During the interphase, the same like in mitosis, chromatin fibers replicate and duplicate, of course, in numbers. So the same is centrioles. Then next stage, prophase, is nuclear membrane disappears, and the chromatin fibers condense into chromosomes. So we have two V-shaped identical sister this V-shaped chromosomes. They already produce X-shaped chromosome, and because they have X-shaped chromosome to separate them from previous, okay, this sister V-shaped structures, they call chromatid. It is chromosome actually, but change name to chromatid, because we have X-shaped chromosome, which is from two sister chromatids, chromosomes, chromatids. Name chromatids just to separate from chromosome. Then metaphase 1, they all these tetrads, they also produced what they group together into tetrads. They group together into tetrads, and then these microtubules, they pull these X-shaped chromosomes to opposite side of the cell, and then these chromosomes, they break. I don't know. They just get into the chromatin fibers, and around these chromatin fibers, nucleus, envelope of the nucleus appear. So we have two daughter cells, which are not identical because genetic information is different, and we have a centrosome in every of the cell. Okay. So okay, let's go. The sister chromatids are no longer identical because of the allele exchange, this crossing over, and that happens during crossing over. Then spindle fibers disappear and nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs and meiosis 1 ends up with two identical different daughter cells. Every cell contains only one set of X-shaped chromosomes consisting of paired sister chromatids. Both cells now enter the next stage, meiosis 2. However, unlike meiosis 1, DNA does not replicate before meiosis 2 begins, so there is no interface. But yet centrosomes duplicate. We have one centrosome in every half of the cell, and they duplicate again. In prophase 2, the nuclear membrane again disappear, and these fibers grow from the two sets of centrioles. And during metaphase 2, the chromosomes in each cell line up again at the equator and attach to spindle fibers from both poles. During anaphase 2, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they are called chromosomes again. So they were chromatids, sister chromatids. Actually, remember that these are chromosomes actually. And chromatid is just name to separate them, to distinguish them from X-shaped chromosome, which and X-shaped chromosome is dyad. It is it consists of two sister chromosomes or two sister chromatids, and we call it chromatids. But then they separate and they change name, chromosomes. Okay, now they are V-shaped chromosomes. Finally, during telophase 2, the spindle fibers again disappear and nuclear membranes reform, and so then the cytokinesis occurs in both cells. So you see that we have in mitosis, we have two V-shaped chromosomes, and they produce X-shaped chromosome from two sister chromatids. We change name from chromosome, V-shaped chromosome to chromatid, and they are connected about in the middle with centromere. Centromere, it is some kind of glue which keeps two sister chromatids together. Okay? And in meiosis 2, centrosomes, these fibers, microtubules, they are connected to every sister chromatid from one centrosome and other centrosome, different these microtubules. So anaphase 2, it is the same as anaphase in mitosis. So we have separation of the sister chromatids, but because there was no interface, there was no replication of number of chromatin fibers. We have now only half of the genetic material. So they separate, and we are getting daughter cells which are not identical to each other. So here, sister chromatids separate. Now we call them the chromosomes, and then that's in telophase membrane appears between the two halves of the cell, and cytokinesis happening. So here it is cytokinesis producing in total 4 daughter cells with different genetic material. Okay, we have that interkinesis. We can call it interkinesis, but in difference from mitosis, there is no replication of these chromatin fibers inside the nucleus. Only centrosomes are duplicate or replicate. And then membrane disappears. These X-shaped chromosomes migrate to the center. They get connected to microtubules, and eventually, when they are arranged around the equator of the cell, microtubules pull them out and separate daughter chromatids. And now we have these chromatids, they turn into chromatin fibers. Okay, the chromosomes are no longer condensed. They exist as chromatin fibers, and nucleus envelope forms around these chromatin fibers. So we have 4 daughter cells. Okay, summary. Meiosis 2 ends with 4 genetically different haploid daughter cells, each containing only one set of chromosomes. Meiosis begins with a diploid cell and produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has 2 stages of cell division called meiosis 1 and meiosis 2. During meiosis 1, homologous chromosomes separate to produce 2 cells, each containing chromosomes in the form of paired sister chromosomes, or this X-shaped chromosomes. In meiosis 2, the sister chromatids separate in both cells and becoming individual chromosomes. So chromatids separate, and they became like these V-shaped chromatids. They separate and they become chromosomes, or actually they get converted into chromatin fibers. Cytokinesis of these cells produces 4 genetically different haploid gametes. Okay, if we look microscope, we can see interface, which is always happen before mitosis and meiosis. And during the interface, chromatin fibers, which are just like spaghetti inside the nucleus, they replicate or they duplicate number. And next, what is prophase, what is different from mitosis, they produce not only X-shaped chromosomes, which are 2 sister chromosomes, 2 sister chromatids. Chromatids change of the name from chromosome to chromatid. But they have tetrad. Tetrad, it is all homological, similar chromosomes. They produce, they pair and produce 4 this chromatid, or it is 2 combination of 2 X-shaped chromosomes. But they stay together. So we have 4 V-shaped chromatid, 2 V-shaped chromatids or sister chromatids, and altogether 4 chromosomes. We just remember that it is all chromosomes. And next, in metaphase, these tetrates, they migrate to the equator between centrosomes, here centrosomes, and fibers appear. And then in anaphase, these fibers pull these X-shaped chromosomes to each other, and then we have separation of these tetrates into diads into these X-shaped chromosomes. This anaphase 1. Okay, complete separation, and then these fibers disappear, and then all this X-shaped, V-shaped, and X-shaped X-shaped chromosome and V-shaped chromatins, they form chromatin fibers. Again, chromatin fibers and nuclear envelope appear around this chromatin fibers. Okay, so you see this, we don't see any structure. This is now chromatin fibers. Next, prophase 2. Prophase 2, it is in every this daughter cell. In every daughter cell we have again this appearance of X-shaped chromosomes, but in difference from mitosis or prophase 1, there is no replication of the chromatin fibers. So we have diploid cells with number of chromosomes for humans 46. And what is happening then, crossing over, a swapping of genetic information between them, these chromosomes. Okay. Metaphase 2, again, these chromosomes, X-shaped chromosomes, they migrate to the middle of the cell, and fibers get connected to them. And then every V-shaped chromatid, which is actual chromosome, is separated and pulled and moved to the opposite side of the cell during the anaphase. Eventually, telophase, we have fibers disappear. These chromosomes also form, they degrade, or I don't know, like disentangle into, gets not condensed. They were condensed in chromosomes, but now they turn to the chromatin fibers, and we see no structure of chromosomes. We have chromatin fibers inside nuclear membrane. So nuclear membrane appears, these fibers, microtubules, disappear, telophase 1. And eventually we have 4 daughter cells due to cytokinesis, so a new membrane appears. And we have, in principle, we can see here some chromosomes, but okay, that's I don't understand much, but okay, again, maybe these chromatin fibers then again condense into chromosome. It looks like, so due to the picture we have appearance of chromosomes, but number of chromosomes is haploid. It is half from the previous number of chromosomes. So if we humans have 46 chromosomes altogether, then we have 23 chromosomes in every haploid cell. Okay, that's all. Okay, I highly recommend to download presentation and also text file, because to remember all these names, which sounds a little bit similar, like centrosome, centromere, and other chromatin, chromatid, and etc. It's not so easy, so I highly recommend to read the text file and maybe to try to remember what is what.